Hi everyone. So we've just about made our way through cardiac physiology. We've considered the pacemaker and non-pacemaker action potentials. We've considered the physiology of muscle contraction, and we've begun to relate the electrical function of the heart to the mechanical function of the heart. But what we need to keep in mind is that all of the electrical function and mechanical function is necessary in order to pump the blood out of the heart, and in doing so, pumping oxygen throughout the body. So now we're going to take a little bit more of a global perspective and consider the cardiac cycle. The cardiac cycle is the start of one heartbeat to the start of the next heartbeat. And just a couple important words for terminology. Systole refers to contraction, and diastole refers to relaxation. So when we talk about things like EKGs, we have to remember that we're talking about depolarization and repolarization. But as we talk about the cardiac cycle, we're talking about contraction and relaxation, so electrical versus mechanical function. So we'll start the cardiac cycle with the heart completely at rest. And when we consider the cardiac cycle, we must consider both what's happening in the atria and what's happening in the ventricles, because they're not necessarily the same at any given point in time. So the initial starting point, the heart is completely relaxed, and we have atrial diastole and ventricular diastole. And during this period of time, since the heart is relaxed, the AV valves are going to be open and blood is returning to the heart from both the pulmonary circulation and the systemic circulation. And as the blood returns through the heart, the blood actually is going to travel directly through the atria into the ventricles and start to accumulate in the ventricles. Now, in order to consider this more thoroughly, let's just relate it quickly to what's happening in terms of the electrical piece to prepare for the mechanical piece. So first we have SA depolarization. The pacemaker cells depolarize, and then the positive charge travels through the gap junctions to conducting cells, and then through the conducting cells into contracting cells. So all of the atrial contracting cells will become depolarized. Once the atrial contracting cells become depolarized, we will travel sort of through that non-pacemaker action potential and get to the point of calcium entry. And during calcium entry, we will have atrial contraction. For the purpose of clarity, both atria contract at the same time, and they both push the blood through into the ventricles at the same time. So initially, during atrial diastole and ventricular diastole, we have some accumulation of blood in the ventricles, and in fairness, some accumulation of blood in the atria. But about 70% of the total blood that's going to come into the heart is going to travel directly into the ventricles. And about 30% of the blood is going to collect in the atria. And then as the atria contract, we have atrial systole. The atria are going to actually squeeze the remaining 30% of blood into the ventricles. Now, let's also consider what's happening sort of simultaneously. So while the atria are contracting, the ventricles are depolarizing. So we know that the conduction passes through the AV node, and we'll have initially slowed depolarization for the AV node because we want to allow for time for the atria to contract. And then the electrical impulse is going to travel. So positive charge is going to travel from the AV node through the conducting cells of the ventricular electrical pathways, and then eventually to depolarize all of the ventricular conducting cells. And once the ventricular conducting cells have been depolarized, we're actually kind of past the point of atrial systole, and we're about to begin ventricular systole or ventricular contraction. So the atria are in diastole, the ventricles are contracting. As the ventricles contract, they're going to push back on the AV valves. So the AV valves are going to close so that no backflow of blood can travel to the atria. And then once systole is complete, once contraction is complete, the blood is essentially going to be pushed out of the ventricles, and then it will travel systemically. It's also important to note that while the ventricles are contracting and the AV valves are closed, there's still continuous flow of blood, and so therefore there's going to be some blood flow that's going to come back and accumulate in the atria. And so that atrial blood, once those AV valves open, will either be able to pass through at that point or will pass through with the next contraction. And this cycle is going to continuously repeat. So this entire page describes one cardiac cycle, and when we repeat it over and over, we essentially have continued heart function. All right, so as we talk about the cardiac cycle, there's a few more important words that we need to consider. And so let's sort of take a step back and start where we have ventricular diastole and atrial contraction. So atrial systole, the atria are pushing the blood into the ventricles, and right at the point when there is all of the blood that the atria have the ability to push into the ventricles, we can measure the volume in the ventricles, and we call it the end diastolic volume. So the end diastolic volume refers to the amount of blood that is in the ventricles at the end of ventricular diastole. So right at the end of atrial systole and at the end of ventricular diastole. So when we talk about these volumes, because there's going to be a couple more, let's just remember that they belong specifically to the ventricles, and hopefully that will keep things making sense. So now we fast forward a little bit. The ventricles are contracting. They push the blood out into the pulmonary and systemic circulation, and then the amount of blood that's pumped out in a single heartbeat is actually called the stroke volume. So it's just the terminology that we use to describe the amount of blood that's pumped out of the heart with one contraction. And then the heart is not 100% efficient. So we have a certain amount of blood that collects in the ventricles at the end of ventricular diastole after atrial systole, and then we pump out some of that blood, but we don't get to pump it all out. And so what we have left over is called the end systolic volume. So the amount of blood in the ventricles after the ventricles contract and expel all of the blood. And also this refers to the end of ventricular systole. So we're always going to keep the ventricles in mind. And if we wanted to consider these numbers in terms of a ratio, we would consider something called an ejection fraction. And so the ejection fraction is basically the stroke volume divided by the end diastolic volume. So the amount of blood pumped out of the heart divided by the total blood, or compared with the total blood that was available in the ventricles to be pumped out. And so ejection fraction, as we said, is not 100. It's not 100% efficient, but a good ejection fraction would be probably around 60%. And when patients have cardiac damage from various disease processes, but for example myocardial infarction, if the heart cells die and the pump function is impaired, then the ejection fraction is going to be reduced. So that brings us to an important equation that we need to consider, and this equation is heart rate times stroke volume equals cardiac output. So the abbreviations are here, and basically heart rate is the number of times the heart beats per minute. Stroke volume is the amount of blood that's pumped out of the heart in a single heartbeat. And so when we multiply them together, cardiac output is basically the amount of blood that's pumped out of the heart each minute. And cardiac output is a really important term. Even though it's not something that we necessarily are going to directly measure on a regular basis, we can consider it just in terms of, from a theoretical standpoint, if cardiac output is reduced, that means that less blood flow is arriving at peripheral tissues, which means that less oxygen is arriving at peripheral tissues. And so therefore we have the opportunity for the peripheral tissues to not be able to produce enough ATP, to start to become damaged, to start to become angry, and things like that. So the heart is essentially just supposed to make sure that we maintain our cardiac output. And when we experience various problems such as cardiac dysrhythmias, we always want to keep in mind whether or not it's affecting cardiac output, because when it is, then it's cause for concern and probable intervention, and when it's not, it's more of sort of a recognition, but not necessarily something that we're going to fix. So let's just consider this equation briefly. So if we were to increase the heart rate then and maintain the stroke volume at a constant amount, then we would increase the cardiac output. And similarly, if we were to maintain heart rate but increase the stroke volume, we would increase the cardiac output. And I think what follows is quite obviously, if we were to increase both the heart rate and the stroke volume, we would also increase cardiac output. But we're going to talk a little bit more about how that gets complicated later on. And then just for the sake of comparison, if we were to increase the heart rate but decrease the stroke volume by the appropriate ratio, then we would maintain our cardiac output. So in order to consider how cardiac output changes, we need to consider both how heart rate changes and how stroke volume changes. Generally, heart rate is going to change in response to neural innervation or hormonal changes, and stroke volume will change in response to changes in the end diastolic volume or the end systolic volume And we'll talk about exactly how that can be shortly. So let's start by talking about how we change the heart rate. So we've said before that the cells of the heart possess the property of automaticity, specifically the pacemaker cells, but it's still fair to say that we have sympathetic and parasympathetic innervation, and that neural innervation allows for changes in things like heart rate. So we have our sympathetic neurons that are going to come off of the spinal cord between T1 and T12, and I'm not really meaning to show you two independent neurons here, but I just want to make this hopefully be a little simple. So our first neuron, the preganglionic fiber, is going to release ACH, which binds to the nicotinic acetylcholine receptor, and then the second neuron is going to release norepinephrine, which is going to bind in the heart to the beta-1 receptor. So here's our normal cardiac action potential for the pacemaker cells, and we know that we have entry of sodium and entry of calcium. And then we'll just draw quickly a pacemaker cell. So in the membrane of the pacemaker cell, we have a beta-1 receptor, and that beta-1 receptor is going to be attached to some G proteins, right? So G protein coupled receptor. And when the beta-1 receptor is activated, we're going to get a cascade of reactions that will generate a second messenger, which is cyclic AMP, and that's going to cause another series of events that ultimately will allow for opening of the sodium channels and the calcium channels that are present in the pacemaker cells. So specifically the hyperpolarization activated sodium channels, and then the calcium channels that are going to allow for faster depolarization. So let's just draw another graph on the same graphs of the action potential, showing the difference for an action potential that has had some sympathetic stimulation. So we get faster entry of sodium, which means that we're going to actually make the slope of the line steeper. And so over the passage of time, we're going to get we're going to arrive at threshold more quickly. And then when we depolarize, we're going to get faster entry of calcium. So faster entry of calcium means that we're going to get faster depolarization, meaning that the cell in turn is going to be stimulated more frequently, and ultimately heart rate is going to increase. It's of course important to remember that epinephrine, norepinephrine are normally what's going to stimulate the beta-1 receptor. Norepinephrine comes from the neurotransmitter, epinephrine more as a hormone, but also any beta-1 agonist is going to mimic these effects. Now also consider the parasympathetic neurons and parasympathetic innervation of the heart. So again, these come from so we have our brain spinal cord. This is vagus nerve, and it actually comes from kind of the base of the medulla. So it comes from the brainstem. And again, we have our preganglionic fiber that's going to release acetylcholine, which binds to nicotinic acetylcholine receptors. And then we'll have our postganglionic fiber, which is going to release acetylcholine. And this time, the acetylcholine is going to bind to the muscarinic acetylcholine receptors. So again, we'll draw our action potential. This is our normal, the red line. And then we'll draw our pacemaker cell. So essentially this could be the same pacemaker cell, and we're going to just add some components that allow for parasympathetic stimulation. So here is our muscarinic acetylcholine receptor. And the muscarinic acetylcholine receptor is also attached to G proteins. And one of the subunits of the G protein, actually, when this muscarinic acetylcholine receptor is stimulated, is going to detach and actually allow for the potassium channels to open. So the effect of opening these potassium channels is that we're going to get greater potassium efflux, and so therefore we're going to have hyperpolarization. So as we pass through phase 3, we're going to actually lose more potassium than we would without the parasympathetic innervation or stimulation. And so there's actually going to be more potassium loss, brings us further from threshold. And the result of that is that with normal sodium entry, we're going to actually take longer to get back to threshold, which means that there's going to be a delay before the next depolarization, which essentially means that the heart rate is going to slow down. All right, so now let's consider the effects on stroke volume. So back to these values that we were considering before, the end diastolic volume, the most amount of blood that ends up in the ventricles after atrial contraction, minus the stroke volume, the amount of blood pumped out of the heart equals the end systolic volume, the amount of blood that's left over. So if we wanted to modify the stroke volume, we could either modify EDV or ESV. So EDV is affected by a couple of things. One is filling time. So the longer the ventricle is relaxed, the more time there is for the blood to fill up in the ventricle. And so if we were to, for example, there are a lot of different ways that we could spin this and consider the events or the results, but we're just going to pick a couple of examples to just work through. If we were to increase the heart rate, then the result is that we're going to reduce the filling time. So faster heart rate means that the contraction is occurring more rapidly, which means there's less time for the blood to fill up, which means that you're going to reduce the EDV, and a lower EDV means that you're going to eject less blood, so a lower SV. The second thing that could occur is change in venous return. So venous return refers to the amount of blood that returns to the atria from venous circulation. So if we were to, and again, we could kind of talk about what happens to cause a change in venous return, and conversely what venous return does to our other numbers. So for this example, let's just say if we were to increase the venous return, more blood comes back to the heart, then essentially we're going to push more blood through, so we have an increase in EDV, which in turn will increase the SV. Now let's kind of fast forward a little bit. So we have ventricular contraction. We're going to push the blood out through the pulmonary trunk and the aorta, and then what we're going to have left is the ESV. So let's consider the things that can affect the ESV. Number one is preload. So preload refers to the degree of stretch of the cells of the heart as a result of the amount of blood that returns to the heart from venous circulation. And so in a way, this is also dependent upon the end diastolic volume. So for example, before we said that if we increase the heart rate, we would decrease the filling time, which decreases the end diastolic volume, which decreases the stroke volume. So decreases in stroke volume is going to reduce venous return, which in turn is going to reduce preload. The next thing we could consider is the contractility of the heart. So the contractility of the heart refers to how much the heart is able to squeeze and push the blood out. So very specifically, it's the amount of force that's produced during contraction at a given preload. So from a specific amount of blood that's coming back to the heart at a particular time, it's going to require a certain amount of force to produce a contraction and to squeeze that blood out of the heart again. If we were to reduce preload, then we would reduce contractility. And conversely, if we were to increase preload, we would increase contractility. And then lastly, we'll consider the afterload. So the afterload is basically contributed by the blood pressure and how clamped down things like the aorta and the pulmonary trunk are. So by definition, afterload is the amount of tension that's required to open the semilunar valves and eject blood. So if you think about during ventricular contraction, initially everything is kind of closed and the ventricles are just squeezing and then increasing the pressure, and then ultimately what will occur is that rapid increase in pressure is going to cause those valves, it's going to just kind of overcome those valves and cause those valves to open. So if we have a reduced contractility, then we have a reduced ability to overcome the afterload. And if we can't overcome the afterload, or the afterload was to increase, then ultimately we're going to reduce the stroke volume and reduce the end systolic volume. So again, let's just remember that our primary goal is to maintain an adequate cardiac output. We want to perfuse the tissues and deliver adequate supplies of oxygen. And the bottom line is we have to be able to make ATP. So as long as we have ATP, we can power the sodium-potassium pump. But if we don't have enough ATP, we can't power the pump. And so after we exchange all of our ions, we aren't going to be able to move them back where they belong in the original places. And ultimately, we won't be able to generate any additional contractions. And so it's gonna just become this progressive problem that may not be recoverable. All right, so we've considered the cardiac cycle as well as some of the factors that are going to affect cardiac output, including heart rate and stroke volume. So that is pretty much it for cardiac physiology. There's still a little bit to do in terms of what happens in the blood vessels and what happens with blood and some of the peripheral things. But in terms of the heart, we've finally made it. So I hope you enjoyed the video, and thanks for watching.